title. Get ready. Press it. Nice.
Hello, my name is Wakana Watanabe. I'm a third grade student in Kyoto University. Today, I would like to talk about what I can see when using mid-infrared spectroscopy on low soy milk. Tofu will appear in this content. Tofu is made of soybean, and this is popular food in Asian countries. Do you know what makes tofu harden? It's betaine. Betaine is a kind of coagulant, and this is consumed by phytate. Phytate is a substance related to the solidification of tofu. This graph shows the relationship between the amount of phytate in soy milk and the hardness of tofu made from that soy milk. You can see that the less phytate, the harder the tofu. Therefore, phytate serving is very important in the production of tofu. To conduct the experiment, first make 3 types of soy milk. There are low soy milk, phytate added soy milk, and dephytate soy milk. Blue is low soy milk, and red is phytate added soy milk made by adding phytate to low soy milk. Green is dephytate soy milk made by refusing phytate from low soy milk. From here, we will make some samples for infrared spectroscopy in 2 ways. The first is method A. In this method, phytate is separated from low soy milk and diluted with straight buffer to make solutions of various concentrations. Method B is to mix phytate added soy milk and phytate-free soy milk in various ratios. As a result of this work, samples were made in 2 ways, A and B. We measure this by infrared spectroscopy to determine the concentration of phytate in soy milk. You try to investigate the concentration of phytic hydrochloride in low soy milk by IR absorption spectroscopy. By comparing phytate and low soy milk, absorption was similar and could not be compared. A is low soy milk, and B is soy protein, and C is soy saccharide, and this is phytate. And you can see 1100 is here, similar absorption. In response to this result, you work to completely separate phytate from low soy milk. To prepare insoluble phytate, calcium chloride was added at pH 11.5 to 12, which minimize phytate solubility. The phytate precipitate here was dissolved in straight buffer, and infrared spectroscopy measurement was performed again. IR absorbance at 170 per centimeter over each solution was used for the measurement of phytate. The absorbance of phytate was converted to the content in low soy milk. The calibration of the absorbance against phytate content is shown in this figure. This means that this procedure can be used to measure phytate. Comparing the concentration of phytate obtained by a chemical method other than infrared spectroscopy with the concentration obtained by this method. Infrared spectroscopy is about as accurate as other methods. Finally, I talk about discussion. Previously, HPLC and iron precipitation methods were used to investigate the relationship between torque hardness and phytate concentration. However, these methods are too time-consuming. In this experiment, two methods were used to determine the concentration of phytate in soy milk. As a result, our precision sufficient for practical use was obtained. Using the infrared spectroscopy investigate, this time we can investigate the relationship as accurately as possible in a shorter time. My presentation is finished. Thank you.
Okay, any question and suggestions, comments? Anyone? Don't hesitate. Go ahead. Today, not so much enough time because the 4 people presentation. Why not? If no one, show us your presentation. I have many things to suggest you. The first, as I mentioned last time, you remember? You need to show us the page number. You forgot it. Okay, do it. And you know how to do it. Again, I'll tell you, all of you, don't forget. For example, if 12 pages, this way, and 2, 12, 3, 12, like that. This would be good to communicate through this PowerPoint. And that's very small letters. We can't see. What's that? From here, fine. Why? No one can read that. That's a very big problem. So you like to say only HPLC? What's that? What this? Very few information. more some letters necessary, I think. And what's conclusion? Conclusion is important. No conclusion. That's a problem. Okay, next time. So many things I have something. Anyway, from you. Any suggestion, any comments, okay. Don't hesitate. Why not? Okay, Higashi-kun.
Thank you for your presentation. Could you explain this graph? What's the difference between white circle and black circle?
Thank you for question. And this white circle was obtained from the mixture of 2 soy milks. and black circle was obtained from this method, and calcium chloride add other calcium chloride to soy milk, and to make completely separated soy, complete diffused phytate from soy milk. That method make black circle result.
So it is better to put the explanation. That's right.
Thank you. I'm sorry.
Okay, more explanation.
Yeah.
Okay, from Kyoto. Next would be Tsukuba. You are ready to give her the question?
Just one minute, please. Hello. Thank you for your presentation. I didn't see the references. Did you do this research by yourself, or did you get article from somewhere?
Thank you. And this presentation is from another person's 研究ってなんですか experiment, and this is from rapid measurement of phytate in raw soy milk by mid-infrared spectroscopy from Takahiro Ishiguro, Tomotada Ono, Katsuhiko Naka-san to Shigen Tsukamoto and Shinji Shimada. And I have should write this presentation, but I didn't do that. I'm sorry for less information.
Okay, thank you.
Hello, thank you for your presentation. I just have a few comments regarding your presentation. One is you presented in the introduction, but to me it is not very clear. What is it you would like to, or the authors would like to have, harder tofu or soft one? Remove completely phytates, or do they want to control the concentration so that they can adjust the hardness?
I'm sorry, I can't understand. Sorry, please say again.
Okay, so what is it that the authors were aiming to do? To remove completely phytates from the soy milk?
Okay, the first method cannot completely separate it. So you cannot see the difference of absorption between phytate and soy milk. So what is caused by phytate and what is caused by other contents in soy milk, you cannot see. you cannot understand. So, it is possible to separate completely phytate.
Okay, may I? You see, basically, we are talking about proteins, I believe, also. You will have them coagulated, but there's no mentioning about how do these phytate and the proteins, is there any relation between them? How do the phytates affect protein coagulation, and then you may have harder tofu? It was not mentioned.
Yeah, yes. Sorry, I cannot understand that point, and I'm sorry, I cannot understand.
Yeah, that's okay. This is more a comment. Just about your presentation itself, slides. Can you please go back one slide? Go back one. Yes. You see, this slide is very difficult to understand because there is no information. You should write clearly which compounds and how did you do the analysis, which equipment was used. Someone who looks at the slide cannot understand. It should be easy to understand only by looking at the slide.
Yeah, okay. I'm sorry. Thank you.
Yes. Yeah, just one comment. Thank you.
Yeah.
Maybe Taiwan, please. Time to move to Taiwan.
Yes, any comments from our NTU students? Yes, please.
Thank you for your presentation. I just want to ask what's the relationship between the Python and the raw soybean milk, and what caused the hardness of the tofu? Thank you.
Relationship between phytate and tofu hardness.
The relationship between the phytate and the raw soybean milk, and what caused the tofu's hardness? Yeah.
Next page. Phytate consume betaine by making phytic chloride. So many phytate is included in tofu if many contained. That tofu, I'm sorry, no. Many phytate is contained in raw soy milk. That soy milk, the tofu from that soy milk is softer. Not enough. What should I explanation?
Okay.
I'm sorry.
Okay. Thank you.
I'm sorry.
Okay. That's fine.
Any more questions or comments? Okay, please.
Thank you for your presentation. I just want to ask, what was the objective of the presentation? Like, what was the main goal or the focus of the presentation?
What's the objective of this presentation?
Yes, yes.
Okay. I want to talk about phytate between relationship between phytate and tofu hardness, and because this is previously that relationship is discussed by HPLC and iron precipitation and other methods, but this method is time consuming, so not useful in practical scene. So I want you want to know and investigate that faster methods to know phytate concentration in soy milk, and this inflated spectroscopy is faster than another method. So the objective of this presentation is to introduce faster methods to know relationship. To know phytate concentration.
Okay, thank you.
Thank you.
Here, I just have a small comment for you on your last slide, please. Last slide. The very last slide. Yes, on this slide you have symbols of the solid circle and empty circle. It's better to put a legend to say what is the solid circle and what is the empty circle. Just a suggestion for your future presentation. You know what I mean? You have two symbols on this slide. One is a solid circle, one is an empty circle. And it's better to put which is which. Okay. Okay, that's just a suggestion. That's just a suggestion.
Okay.
Thank you. So that's all from NTU. Thank you.
Okay, thank you very much. Your discussion and many suggestions. That was Tanabe-san. Okay, you may include next time. Okay, thank you very much. Next would be the Hirono Narumi-san. NTU and Tsukuba, you can see a material?
Yes, we can, from Tsukuba.
Yes, we can see
Okay.
it from NTU.
Okay, thank you very much. Okay, next presenter would be Hirono Narumi-san. And the title. Okay, title was changed. It was eggplant, but now the grow of firefly. Totally different. Huh? Okay. Okay, if you get ready.
And the title I said before was unexpectedly difficult, so I changed the title. But it became a content that wasn't very relevant to agriculture. I'm sorry for that. I investigated the luminescent substances contained in firefly. This is the timetable of contents. The reason I chose this theme was because I had an experiment on the sea firefly light emission mechanism when I was a junior high school student. Then I researched about it for 5 days at Kyoto Koreisen University. So I chose this theme. I measured the quantitative data and reproduced the light in the test tube. Then I will talk about how fireflies grow. There is a luminous organ. This that looks yellow in the area close to the bottoms of the fireflies. Among them, there is a substance that emits light called luciferin and enzyme called luciferase that helps emit light. So these two substances react with each other with oxygen in the body to emit light. This luciferase varies depending on the firefly. Therefore, in addition to generally known yellow-green light, there are various light colors such as yellow or orange. This is about the luciferin. This luciferin is a generic term for substances that emit light when oxidized by luciferase. This is classified into the following 5 types. Also, the firefly light is not hot. The enzyme luciferase is produced by living organism in the body that is a protein that promotes chemical reactions efficiently. About 40% of the energy changes to light, and it does not give off much heat. Among bioluminescences, fireflies are said to have the highest luminous efficiency. By this function, it does not get as hot as light bulb. Next, I will talk about bioluminescence reproduction experiment. I researched actually when I was a junior high school student. This is the method. First, I put the dry firefly in a motor and greened it and add various aqueous solutions in the dark room. And then I measured the luminance. It is the result. When the experiment was carried out by changing the type of water under constant temperature, soft water grew more strongly than hard water. Moreover, when the same experiment was conducted by changing the amount of potassium, the water containing more potassium was more intense. Furthermore, when the experiment was conducted while changing the temperature of the water, it was the strongest light at a normal temperature. This is the conclusion of the experiment. It became clear that based on experiments focusing on water components, the fact that the concentration of potassium in seawater is greatly related to the light emission of the sea fireflies. It was found to emit light most easily in seawater at 20 Celsius. We can narrow down the habitat of sea fireflies by this experiment. This is how to synthesize the spring. This is found by the professor of Chubu University. The method is to put chemicals, benzoxide, and cysteine in neutral water and stir them at room temperature for 3 hours. Trace amounts of luciferin are synthesized in this process. Even in the science room of elementary schools, it is possible to make a light-emitting substance. It is presumed that this is expectation. It is presumed that 100 million years ago, a similar chemical reaction occurred in the body of firefly ancestors, and luciferin began to be synthesized. Luminescence by luciferin is also used in testing reagents. Unlike other fluorescent materials, luciferin has the feature that it emits light without a light source as long as it has ATP. The discovery of a simple synthesis method may not only reduce the cost of testing reagents, but may also lead to new applications. That's all. Thank you for listening.
Okay. Any suggestion, comments, question? Please, go ahead. With the mic.
Thank you for your presentation, and I forgot the slide number, but please show me the when you show us soft water or hard water in your experiment. This one. In this experiment, what soft water means and hard water means?
Soft water is nansui. Hard water is kousui.
How did you get that?
How did you? I tried the concentration of potassium, but I didn't do experiments about this in real experiment. Sorry.
Okay, I got it. Okay, that is, thank you.
Okay, thank you. Anyone else? Please, anyone. One more. No one? That fireflies light is fluorescence? Fluorescence? Fluorescence?
蛍光?
Maybe not.
Maybe. Yeah, 蛍光. Yes.
Oh, that's right. If so,
Maybe.
excitation wavelength is? To get the fluorescence, we need the excitation wavelength, light, and we can emit light emission. But that is not fluorescence.
Maybe.
I don't know. Yeah. Anyone? Okay, no one? You saw? Okay.
Thank you for the presentation. So if I understand correctly, you're saying that by using these fireflies, by using the molecule Lucifer. Yeah, we can use that to substitute for luminescence or fluorescence experiment in the lab. Is that what is that one of the application for it? So, okay. Yeah. Okay. No. Thank you.
Right.
Okay. Yeah. Third grade students, not enough knowledge, but try to do learn more, especially English, some technical terms. Anyway, that's all from KU and Tsukuba, please.
Thank you for your presentation. Do you know other substances that fireflies or glow more strongly?
Other than this phase?
Yes, other than potassium. Did you study with soft water and hard water, right? Yeah.
What conditions to be more stronger?
Do you know, in your research, you studied with soft water and hard water? I think there are other substances in water.
For example, mineral or calcium.
Yes.
I didn't research about it.
Thank you. This is maybe just suggestion. Thank you. Thank you for your presentations. I'm sorry if I just missed your explanation, but you said 40% of energy consumption is the most efficient, but among what the 40% is most efficient? Comparing what it's most efficient?
Maybe most science, in insects or animals, they couldn't emit light so efficiently, but I didn't compare from other animals.
Okay,
So,
thank you.
No.
Okay, thank you.
Hello. I just have thank you for your presentation. Minor questions regarding your presentation. I did not understand well. Actually, one of your colleagues at Kyoto made a similar question. What is the objective here in this study? You mentioned that knowing the luminescence or how fireflies emit light may be important to the agriculture, for instance. Can you please elaborate more, explain the reason why this research, this study is conducted? The objective of this study, how can it be related to agriculture, for instance? At the beginning, you mentioned that this may be important to agriculture.
Relative to agriculture, other than fireflies? Fireflies? This research, maybe this is not relevant to agriculture. So I made a mistake of choosing the titles. So and I think that it is not related to agriculture, but it may. I don't know, but it may relate to agriculture somewhere.
Okay, thank you. Just I was wondering why this was chosen. And a very basic question, can you explain in your slide there is soft water and hard water. What is soft water and hard water?
Soft water? What is it? What is soft water and hard water? Soft water. How soft the water is varied from country to country or conditions. So what is soft water?
Mineral contents?
Yes. They has different mineral content.
Yes, I see. Might not be related to the hardness itself, but the composition of water, right?
Composition?
Yes.
Sorry, I forgot about it.
Yeah, it is fine. Thank you. No more from school. Thank you very much. From Scuba, these are all our comments. Thank you.
Please, NTU.
Okay, due to the time, only one question. Yes, please.
Okay, I just had a quick question. It's about the influence of temperature in the sea, because you said that depending on the temperature, the light will be the intensity of the light will be stronger, more or less stronger. So I just wanted to know what was the influence of the temperature on the light. On the light. Yeah. Right. Thank you.
Temperature. Intensity. Brightness. At the 20 Celsius. いつ時点あら、本当だ。あれ something more intense. Intense. Temperature. It was 20. あとは normal temperature.
You wrote that.
You wrote that?
That's your PowerPoint.
あと normal temperature is from 5 Celsius to 20 Celsius.
Why?
It is 性質 nation.
You don't know, just
I don't know why, but it is 性質って nation. Property. It is a property.
Okay, thank you.
Okay, this is the end of the Q&A from NTU. Thank you.
Thank you very much. Discussions. We'd like to move to the next presenter. Will be Tsukuba, please.
Is it possible to make the screen face to camera?
Okay, we have 4 people.
Can we move the screen to face to camera?
This.
Only one question because of the time limitation.
Can you see the signal of the light?
Thank you.
Thank you.
We also okay.
So I will start my presentation. So good morning, everyone. My name is Brian Ferranda, and I'm a third-year agrobiology student from University of Tsukuba. So I'm going to present about crop signaling as a novel crop recognition technique for robotic weed control. So to give you a brief background, you can see here a world population graph from 12,000 years ago until 2100. And as you can clearly see, from the last 200 years, our world population has rapidly increased, and it is not stopping. So what is the main challenge for us? With high population, there will be more people to feed, thus increase in food demand. Most of our daily food is from crops. So to improve this food situation, we need to improve our crop production. Some key factors for crop production would be land, water availability, type of crops that we plant, and seasons. However, one prominent negative factor would be weeds, which are any growing unwanted plant that prevents main crops from growing freely. So why are weeds a negative factor? For one, it reduces crop yield because it competes with the main crop in terms of nutrients from the soil. Another one is the possibility that it can produce negative allelopathic compounds that will negatively affect the crops. So farmers usually, as of now, use herbicide for controlling weed propagation. But some of them, the herbicide is limited for some crops and it doesn't completely eliminate all the weed. So they use manual weeding as a supplement. However, manual weeding is very expensive since they have to hire a lot more laborers to remove all the weed. Another solution would be to automatize weeding. Some of the current technologies used now is like the automatic weeding technologies, like this one, which is a robovator. And however, these current technologies have limitation with depending on the crop type and the spacing between crops and the rows, and especially depending on the abundance of weed in the farm. As I mentioned, high weed density environments is a major limitation for this. During this situation, these machines cannot efficiently remove all the weed completely and carefully remove the weeds without damaging the crops. So the objective of this study is to find a more efficient method for detecting weeds, especially differentiating from the main crop and the weeds. And another thing is to have a weed removal system with the least damage to crops. In this study, they focused on crop signaling, which is a new technique in the field of weed control. This research aimed to apply fluorescent compounds to be able to accurately detect the weeds. Crop signaling techniques are aimed to be safe to humans, machine readable, harmless to the plant, leak-proof to the surrounding soil, and cost-effective. The researchers focused on 4 different signaling techniques, which are as follows: systemic markers, fluorescent proteins, plant labels, and markers. So for the methodologies, I will discuss the 4 techniques separately and share the respective results in the section afterwards. For the first one, they use systemic markers. And the researchers use systemic markers on the plants to label the main crops. So for the case of direct seeding, they use signaling compound and put it inside the outer barrier of the seed. However, for transplanted crops, they put the signaling compound in the seedling plug, which is the one below the transplanted plug. These compounds is very useful since these systemic markers move to the leaves during growth, and thus it is easier to read for the machine when they grow. To maximize effectiveness and retainability of the markers, the researchers made sure that they have put enough amount of the systemic marker in the seed or in the seedling plug, and made sure that the outer barrier structure of the seeds are very strong to prevent leakage. The experiment they did is on romaine lettuce, and they have small transplant flat, which is the first picture at the bottom, number 1, and they put soil and they planted lettuce samples inside it. And during the first leaf stage of the lettuce plants, they put rhodamine B, which is the systemic marker they used. And after more maturation, they have transplanted these plants to a bigger pot. And then where one pot contains one treated plant and another plant without the systemic marker. And they observe it under daylight and UV light. The next technique they use is fluorescent proteins. Usually, most of the scientists, green fluorescent protein is very famous to be able to track gene of interest and indicate successful transformation of target organism, since it emits bright green light under UV light conditions. However, the researchers used anthocyanin gene from maize and put it inside tomato plants. Basically, this anthocyanin gene made the tomato plants appear as purple under daylight conditions. And because of this, the color difference became more evident for distinguishing it between each other. So they changed the gene in the tomato plants. The next one would be plant labels. This one is a bit interesting since they use an instrument, usually a straw, made from plastic, wood, or metal, to be able to identify where the plant is. So like the systemic markers, they planted first the seeds in the transplant flat, which is the bottom picture. And then they also aside from the seed, they also put the straw. And they assume that when the crop grows, its root attaches to the straw. So after growth, they remove that plant to the larger field, which is shown in the picture above, upper right of the slide, and then they assume where the the straw base is, that is where the plant is. And they use multi-imaging system to read where the plant location is, and I will show more later. Lastly, would be, in contrast, would be the plant labeling. Topical markers are used using fluorescent substances, but in this case, they put it directly on the surface of the plant. And there is an automatic system on spraying the plants, which you can see on the upper right of the slide. And it is optimized to release an appropriate amount of the marker so that it will not obstruct on the regular photosynthetic process of the plant. The weeding machine will then release UV light and detect the fluorescent substances on the surface of the plant. Thus, it can already locate where the plant is, and all the surrounding soil would be full of weeds. For the results, for the first technique, for the systemic markers, basically you can see in the picture, in letter A, the daylight condition, there is a small difference. Maybe you will not see it clearly, but there's a small difference with the color. This is slightly purple, which is the one with the systemic marker, and this one is the one without. However, when you put them in under UV light, it is more evident, since the plant with systemic marker would appear as bright reddish orange, and the one without systemic marker would appear violet, dark purple. Thus, the efficient color contrast for machine vision-based reading will be more evident as well. Next one is the fluorescent proteins. Well, the results for this is almost the same as the systemic marker, since it utilizes the difference in colors. However, for this, the researchers think that this is more efficient, especially when the plant is obstructed by a weed, since, as you can see in the upper right corner here, the main crop is actually partially obstructed by the weed, but due to its difference in color, which is bright green, it can easily identify which one is the one with the fluorescent protein and the one without. And the one without is assumed that it will be the weed, which should be removed by the machine. For plant labels, here is the one I was talking about the multi-imaging system. For this is a bit different. So they use 6 different mirrors to be able to see where the straw is. And this is more efficient because if you look at one crop from a specific angle, sometimes it will be obstructed. The location of the plant base will be obstructed. However, using the mirrors, you can see it from 6 different angles, and you will be able to see where the plant is directly. And in the 9 trials, the researchers have said that they have 97.8 success rate during dense weed environment, which is very good already. And you can see in the bottom pictures, these are the plants, and there's a lot of weeds. So as you can see, it's very hard to determine it by just looking. So with the 97.8 success rate, I think it's really good. And the last one is for topical markers, where they use to detect the fluorescent substances on the surface of the plant. As you can see, upon reading of the machine, they can already know where the plant is with its red signal, and the ones without the fluorescent substances would have green ones. Thus, the efficiency of distinction of the crop between the crop and the weed is very high at 99.7%. To conclude my presentation, I would want to make this message that crop signaling has higher accuracy compared to currently widely utilized weeding machineries, with almost 100% positive error rates and almost 100% also of distinguishing between crop and the weeds. And it's relatively more cost effective due to the accuracy that it can provide. So lastly, crop signaling techniques should be developed more to augment efforts in automating farm work so that it would be easier for the farmers and more cost effective. Thank you. This is the reference I use for this presentation.
Thank you for your presentation. My question is if the topical markers are safe for humans to consume.
Oh, regarding that, most of the signaling molecules that they have used are safe. Basically, they're safe. However, for topical markers, I think it's a bit more safe because when they spray the topical markers on the crop, they will not harvest it until the first surface, the leaf outside doesn't fall off. So when they harvest it, most probably the one with the topical marker is already fall off from the crop. So yeah, they don't have the topical markers for consumption.
Thank you.
Thank you, Brian, for your presentation. Sorry to Professor Kondo and Professor Chen. We went ahead asking questions since we were already here. Sorry for that. I just have one short questions to Brian. It seems that for among all the methods that you presented, plant labeling might be maybe the most successful one. And is there any actual usage already in the field? Do you know of any information if it is being already implemented in anywhere? And well, on top of that, just another short question also. I'm worried if you can please explain. Second question is about topical markers. Aren't they washed away? Say if they're just sprayed on the surface of certain plant, then when it rains, it might be washed away or so.
So for the first question, I was not able to see the current commercial use of the plant labels yet. I have tried to search, but then apparently everything is on research, and no farms have implemented on a wider scale. For the second question, the topical mark. Actually, I think that would be one of the limitations, because as in the paper, they have mentioned that the topical marker they used was water-based, and I thought at first that it was better so that they can easily wash off the topical markers, right? But on the condition that it is raining, I think it would be one of the limitations. So I guess they have to research more on having a more safe without having to compromise on the methods.
Okay, thank you. Professor Kondo or Professor Chen, go ahead, please. From Scuba, I believe these are all the questions we have.
Okay, any comments or questions from NTU? I have the questions for you. Since you use fluorescence for detection, could it be possible to do it during the daytime in the real situation? Daytime. Because
I'm sorry, I was.
Because you use the fluorescence technology to detect the weeds. I mean, detect the crops, and so it can be possible to be applied to the operation in the daytime.
In the data, I guess it can. I think it can be used in the daytime since usually they don't really rely on daylight images. They excite the light. They put UV light on the crop. So most of the images that they utilize in detecting where the plant is would be from the UV light exposed images. So whether it's in daylight or in nighttime, I think it can be used.
But be careful about the intensity of the fluorescence is really weak. So that's why I'm worried about the application in the daytime. Okay, thank you. So this all from NTU, so now it's KU.
Okay, anyone?
Thank you for the presentation. I have a short question. It was more with regards to, are there certain crops where this wouldn't work, as in the robot wouldn't be able to detect the weeds or to detect the crops and differentiate from the wheat because either the crops are too high or the base is too wide? Does it work on any crops, or is there specific crops that it works better on compared to others?
Good question. Thank you for your question. Based on the literature I have read, most of the crops that they focus on are leafy vegetable, green leafy vegetables. And then I think it would be harder to apply this kind of method if, for example, it's like crops that grow underneath the ground, because probably it can hit the weeding machine can hit it from below, because they have to remove the weeds, right? So they have to like put a certain material underneath the soil and then go around the plant. So I think if it grows underground, it will also be like disadvantages, or it's hard to predict where the plant is.
Thank you.
Thank you.
Okay, thank you very much. Since we have not enough time, please move to the next NTU student presentation.
Thank you. The first speaker, the last speaker is from NTU. So the speaker is the Jie Wei, please. Do you got it? PowerPoint, KU and UT.
We can see it.
Yes, good.
Okay. Okay, go ahead.
Okay. Hi everyone, and this is my presentation topic is detection of photosynthetic active and water straight by imaging array chlorophyll fluorescence, and the next is the other information and a resource from, and later is and the last is my information and my email address. And in modern time, we relied on technology to develop precision agriculture, not only environment, but also resources. We want to find it and use it friendly and efficiently. So that is a question we want to talk about. When is the irrigation timing is suitable for the plants? And in some area, they don't have enough water for watering the plant all the time. In another situation, for some plants, they are high value, but they are very sensitive to the moisture content. So there's a critical issue: how to know the plant lack of water. So in my above mentions, I give 5 requests for the application. The first is avoid water shortage for plants. And the second is the efficiency of water usage. The third point and the fourth point is we want to observe the plants and get information, but we don't want to harm the plant at the same time. And the last point is we want to get the information in real time, and it can leave time for us to make correct decision. So that's the application of chlorophyll fluorescence. Here are some applications, and later I will talk about how they work and how they work principle and with some data and chart. Okay, here is a picture. When the plant got the light, how will it do? So it can divide three phenomena. The first is photochemistry, and the second is chlorophyll fluorescence, and the third is heat. We want to discuss is about chlorophyll fluorescence and the plant with the relationship with water strains. There are two basic concepts I want to introduce. It can help us to understanding easier for the next content. First is chlorophyll fluorescence. When the plant got the light, the electronic from the high order state to the low order state, the energy gap will turn out form the chlorophyll fluorescence, and that is what we observe above. And the second is cold sky effect. This is an important phenomenon. Look at the picture. We can see in very short time, 100 of a second, the maximum strength arrive the fluorescence arrive at maximum strength. Oh, here we can see, and when the time with the time increasing, we can see the value will get a steady state. It's very important how we observe. So we decide the method to observe it. First, we plan the growth. We plan the seed and make the plant growth. And second, we use fluorescence imaging system to get the image from the leaf. Okay, and third, from the leaf picture and photos, we deal with it, and we want to analyze this from the photo to get some useful information and transfer it to some numerical digit, and help us to make the decision. Here is a spectral analysis. We can see for the 3D scanning, we can see when the excited light at 40 nanometer and the absorbed light at 680 nanometer, the value will get the maximum here. And there are 2D scanning. It can help us understanding easily here. So we use here to the standard, we detect it. So here we decide the procedure. Here is the procedure, our imaging system, and I describe it. For the step 1, we make the plant 20 minute production. Step 2 and step 3, according to the spectrum analysis. So we know we excited by blue light. And step 3, we observe the step 3 is according to cold sky effect. We can know, and step 4, step 3 and step 4 according to the cold sky effect. We can know the maximum value is at 0.2, and the steady value is at 5 minutes. And the last point, we use CCD camera to get the information from the leaves. And here is the photos we take. From the A and the B, we can see the high value, the most maximum value FM, and the steady state fluorescence state FAs. And we use the more important is picture C and picture D. We can make the not only picture, and we can transfer it to the numerical digit. C and D, we can know FN divide C is FD divide FAs, and the D is FM divide FAs. How we use it? We can use transfer the image from RGB and transfer it to the numerical digit, and use this kind of pointer to make us easier understand the situation about the plant. Here is a histogram on the paper, and we compare with these two histograms. For the left side and the right side, we can see the left side is normal watering situation, and the right side is 10-day water shortage. The left side, you can see RFD and FN divided FA is much higher ratio than the right side. And the second picture I want to talk about is you can see the FN and FA gap. The normal watering is much bigger than the 10 days water shortage. And we use our imaging system by the cause effect to test. So we can see in constant water stress, we can see RFT value is with time increasing, and it will be bigger, and it makes sense because you can see the downside. You can see RFD is mean FM minus FAs and divide FAs. FM and FN is with FAs is with the time increasing, it will be the lower. So you can RFD will be much higher. And the B and the photo B I want to describe is with the time increasing, we can see the fluorescence will be decreased with the time increasing. So it can make sense and improve the imaging system is useful. It can be used. And this is my conclusion. We can use this application for quick screening for the fluorescence. And the second is we use FM divide FAs and RFD value to transfer the picture photos to numeric digit, and it can make us to easier judge. And the third is this kind of application can help us to earlier find what happened to the plants, and we can leave more time for us to make decision. And thanks for your listening.
Any questions or comments? Say something.
I didn't understand. Okay. It was just a fast comment on your presentation. I just wanted to tell you that it was good and quite complete. I mean, I found it quite complete and I liked it. So, yeah.
Thank you. Again, sorry, I'm not listening carefully.
So sorry. Yeah.
Pardon.
Oh,
can you explain it again?
I just it was not a question. It's just that I liked your presentation. Oh. You explained well the techniques.
Thank you.
Just comments. Okay.
I have a little. I have a question in your math page, method page. The right picture, the method page. No, the title is method page.
The title of method. Okay. Here.
I don't really know the red in your right picture.
The right picture here. Oh.
I don't know the red and the green meaning.
Okay. The meaning is the red is high value, so you can. It means the detection. You can get the value. The red is mean higher, and the blue is mean lower. It's the strength. Can is it? Okay.
Okay, Professor. Yeah.
Okay. Can you go to the page 14?
14. Okay.
Just the comments. So when you explain the result, if you can put some summary for the result, it makes the audience to understand the key points you want to highlight.
Okay.
You know, because in the figure it contains a lot of information. So if you put some several keywords on the left-hand side, then the audience will understand the key points you want to highlight. Thank you.
Okay, I just a comment for you. Your page number is too small, so you could make it bigger for next presentation.
Okay.
Okay, so that's the end of NTU. How about KU, please?
Yeah, anyone? Any question to Juiche Wei-san? No one? Okay, next time I have to designate. Okay, anyway, let me ask one question, a quick question. When you get the fluorescence imaging, what kind of, how many nanometer or what's the wavelength excitation?
You say the absorb light nanometer?
No, excitation.
400 nanometer. According to the
400,
and we use the spectrum analyzing, we find it's the same. When the excited light at the 400 nanometer for the chlorophyll is the maximum value we can detect easier. For the most easy.
400. That is a very critical. 360, 70, and 400. That fluorescence activities are changing day by day and conditions. Yeah, that is very interesting to me. You are a third grade student?
Oh no, I'm the freshman. A freshman in master degree.
No, you should say
master. Okay, I got it. Yeah, compared with our third grade students, very fluent and very nice presentation, I thought. Okay, that's good. Okay, that's all from KU. And Scuba, please.
Thank you. How about UT?
Thank you for your presentation. Actually, as it was already mentioned, you explained well an interesting topic about image analysis for detecting water stress in plants. I have a comment, actually two comments, general comments, presentation style. One of them being, I have the impression that you use the terms "here is" as you are facing the screen of a computer display that you have there. But I would have preferred if you face the audience, if you talk to the audience, not to the computer. So if you could please, look forward, look ahead, look to those who are listening to you, and try to avoid here is as you are looking to the screen and you are talking to the screen. So talk to us, talk to the people listening. Yes, thank you. Not only to you, but to all the students presenting. And one more general comment also, it was already mentioned today. We do not understand whether this is your own work or someone else's work if you are presenting one article that is already published. So whenever anyone uses any information which is not your own, you did not do it by yourself, the credit should be given to the author. Always mention the reference, not only in your case, but to all the students who presented today also. This is very important. Otherwise, people listening to you, they will suppose that this is your own work if there is no name.
Here is the older information, and here is the page.
I see. Okay, thank you. Anyhow, so all the figures you used are from this article, from this. I see.
Here is I use the device to here just this one. This page is I use device to make data, and the other is from others.
You mean you analyzed by yourself in this case, right?
Yes. Yeah.
Yeah. So it should be made very clear whether it is your own work or someone else's work. Just one suggestion. Thank you very much. This is all from Scuba. Thank you.
Thank you very much. So, we give him big hands. Thank you for the presentation, the last presentation today. So we go to cross today's instruction and presentations. And so next week we're going to have also 4 speakers, and 1 from UT and 3 from KU. So any comments from Kondo Sensei or next week?
Maybe Tira Sensei. Any
no members.
Final?
No, sorry, no.
Members are okay. Members are okay. Okay.
Not the next week. No
this
members.
is okay. All of you get ready. Sugihara-kun, Kim Jun-bin not here today, and Konishi-kun. Okay.
Next week will be
we are okay.
You only.
Yes.
If no more questions or comments, and thank you. So see you. You have something? Okay, please.
Hi there. I have 2 things to announce. The first thing is we create a night group of this joint class, and after this class, go to find your TA and join our night group. Yeah. And the second thing is please give the slide to your TA, and TAs collect them to me, and I will upload to Seba, and you can all students can obtain the slides on Seba. Yes. I appreciate.
So 2 things. One is the TAs are creating a group online, so all the students from 3 universities can communicate each other or discuss each other easily. And second one is all the presentations from students will upload to the Seba system in NTU, and you have a password to download the PowerPoint for your own references. Okay, so thank you. So see you next time. Bye bye.
Okay, thank you.
Thank you very much.